Welcome everybody to the ReSViNET webinar. We are very happy that so many of you are joining us today, this morning, this afternoon, this evening, wherever you are. If you want to participate in the discussion, please microphone or your video for practical reasons, but we will use your questions and answers to have you participate in the discussion. Next slide. So before we start with the webinar, I would like to share our vision and mission of the ReSViNET Foundation. We have the vision to connect global expertise and leadership in order to decrease the global burden of RSV infection, with the mission to support performance of high-quality research, to improve knowledge of RSV epidemiology, and to develop safe and effective therapeutic and preventive interventions. We want to advocate and create awareness for RSV infection, to act as a focal point for effective partnership with all stakeholders, and to combine knowledge and capacity required to enhance the development of novel RSV therapeutics. So why do we do RSV research and education? I'm sure that you all know that RSV is the second cause of death during infancy following malaria. There are 33 million patients, 3 million hospitalizations, and 120,000 deaths, most of them occurring in the first 5 months of life. For this reason, we have started our ReSViNET webinar series to promote knowledge sharing of the latest developments and publications on groundbreaking studies, and to provide education to professionals, specifically also young professionals in the field of RSV. We have had 11 webinars so far with 100 participants on average. We were well evaluated and there was good global representation, and the quality of interaction was well received. So we feel that we are doing something meaningful. Yeah, next slide. So before we move further, I would like to announce a save the date. February 13 to 16 next year, we're going to have our RSVVW meeting 2024 in Mumbai. Actually, my colleagues from the foundation are in Mumbai, and they just informed me that the food is wonderful and the people are super kind. So we really invite all of you to come to India in February. And I can tell you already that one of the chairmen is the speaker of today. Next slide. So I would like to share with you that we have completely renewed our website. A lot of effort has gone into creating a website that is more accessible and easy and intuitive from what we used to have, where you can get the information of the webinars that we're doing, the research that we are following and doing ourselves, and include the patient network, where you can see how patients feel about RSV infection and the solutions that we bring, not only in the developed world, but we also start to engage with patients in the developing world. Next slide. So let's move to the 12th RSV webinar. It's my pleasure and privilege to introduce Barney Graham. Actually, Barney sent me his resume, and I know that he's going to be very upset that I take more than one sentence, but not much more than one sentence, Barney. He started his academic career at Rice University as an undergraduate, and then he moved on through getting a medical degree at University of Kansas to the Vanderbilt University, where he really learned to become an infectious disease expert and a vaccine developer. In 2000, he went to the vaccine research unit at NIH as a founding member. And after his retirement 2 years ago, he went to Morehouse University or Morehouse College, which is basically a black college with the mission to develop people who will lead lives of leadership and service, something that I really admire and I think is of extreme importance to science. He has received many awards and was named one of the world's 100 most influential individuals by Time magazine in 2021. Barney is known for a lot of research in the vaccine field for many pathogens, including HIV, Ebola, chikungunya, influenza, Zika, coronaviruses, including the COVID-19 vaccines, but he shared with me that RSV is and hopefully will always be his #1 child. And that's why we invited Barney to talk about the progress in the RSV vaccines and monoclonal antibody field today. Barney, it's my pleasure to have you here. Thank you so much already. The floor is yours.
Thank you, Louis. And it's a pleasure for me to be here today. I'm looking forward to reviewing some of the new vaccines and monoclonal antibodies we have available or coming available soon. It's been a lifelong dream of mine to see some of these things happening. And I'll just say a word about my new position at Morehouse School of Medicine, because I want to come back to this at the end. That I'm don't have a laboratory anymore. Morehouse School of Medicine is in Atlanta, Georgia. It's very focused on equity, and I'm senior advisor for global health equity. Once we have effective products, this is going to be the major focus for me, at least, is to get them spread to the places that need them the most. Have a number of conflicts of interest. I'm an inventor on vaccines and monoclonal antibodies for those viral diseases, and I'm on scientific advisory boards and consulting for a number of pharmaceutical companies. Won't repeat what Louis already told you. RSV is a virus that infects people throughout the age spectrum, and it's probably best known for its disease in very young infants. And when disease is severe in those children, it's associated with childhood wheezing and asthma, and probably damage to lower airways and lung development that can affect people throughout their life. It also causes severe disease in the frail elderly, and so that has been a problem that's been increasingly recognized. And I think as of last year, when we had an early Northern Hemisphere outbreak in September and October, that much of the world learned about RSV for the first time. And so we have to really connect with them as we bring these new products online. And of course, the legacy of the vaccine-enhanced diseases from the 1960s vaccine trials is why it has taken so long to get to this point. The period of disease, severe disease risk spans from very young infant through about 5 years of age, where at least in the US, this is the leading cause of hospitalization in children of that age. And then it increases again as the elderly become more frail. RSV is an orthopneumovirus in the Pneumoviridae family. It used to be a paramyxovirus, but it was separated out several years ago. It infects the ciliated airway epithelial cells, shown here, sparing the basal epithelium so it can be regenerated, and also infects the type 1 pneumocyte, shown here. This is different than coronavirus, which infects the type 2 pneumocyte that makes surfactant. So the disease pathogenesis is somewhat different, but in the RSV at least, a lot of the disease in young infants is caused by airway obstruction from sloughed epithelium, inflammatory debris, and mucus. And we're going to focus mostly on the F-glycoprotein today. It's one of the 3 major surface proteins of RSV. Be a lot to talk about on these other proteins as well, but F is the one that is generating some of the new products. F has been studied as a vaccine antigen for many, many years, since the 90s. Attempts either using F purified from virus or made in insect cells or CHO cells. There's been several efforts to make an F-based vaccine, mostly tested in adults, but largely so far not as successful as we needed it to be to take it into product development. That changed in about 10 years ago, working with Jason McClellan, a fellow in Peter Kwong's lab at the time, using antibodies that were either identified in the patent database from AIM Therapeutics, Tim Beaumont, or developed de novo from our group in collaboration with a group in Xiamen Medical University, led by Professor Shah. We identified monoclonal antibodies, and these are the FAB fragments that could hold the top of the RSV F protein together in the prefusion form, while the bottom was held together by a trimerization domain, and that allowed us to capture this prefusion form of F and identify a new site of vulnerability, which is attracting monoclonal antibodies of high potency. This F protein rearranges, and this is the prefusion form and the trimeric form and the rearranged postfusion form. And you see this epitope at the apex that we called site 0 is lost when the trimer rearranges. And you can see this in greater detail here when just showing the protomer, a single protomer in the before and after rearrangement. And the reason this is rearranging is to form a heptad repeat that can be the engine that pulls the membranes together and cause a fusion pore and allow cells, the viral genome to enter the host cell. And so these helical domains have to stack on each other to create this peptide repeat. And we'll come back to that concept a little bit later. But the important thing is that this epitope disappears during the rearrangement and no longer attracts the right kind of potent monoclonal antibodies. So making a vaccine with this is not as effective as making a vaccine with this. There's many ways to stabilize the protein. We did it primarily, the dsCAV1 that we'll be talking about today is made by a disulfide bond between 155 and 290, just changing 2 serines to 2 cysteines, and then cavity filling mutations at 190 and 207. But there are other places in the molecule to affect change, and we'll be talking about some of those later. There's a place at the base of this molecule where the stem begins and the head of the trimer comes together in this ring that can be modified to improve stability. And one of the earlier findings was a proline substitution that Hans Longerdick made at 215, which is right at the top of this alpha helix and prevents some of that alpha helical stacking to make that peptide repeat. So stabilizing F in this prefusion form is then what allowed vaccines to be possible. And it also allowed mapping of the antigenic surface of the virus in both the pre and post-fusion forms and demonstrating these new sites where more potent antibodies recognize this apical site 0 or the site 5 that's just below it. And even though some of these neutralizing targets are on the post-fusion form, like the site 2, which would be a palivizumab or motivizumab binding site, or site 4, those antibodies turn out not to be as potent. It also explained why there's two different forms of F on the surface of virions. The pre and the post-fusion form fit perfectly into these EM images, and suggesting that F can flip from the pre to the post spontaneously on the virus surface. And we think this may explain part of what happened in the vaccine-enhanced disease back in the 1960s, when the whole inactivated, formalin inactivated virus was baked at 36 degrees for 72 hours. And when that happens, you kill the virus over about most of it's dead within 48 hours. And we think a lot of that is based on the F flipping from the pre to the post-fusion form, because motivizumab and a dot blot can detect F on the surface of viruses all the way through, but these pre-fusion specific antibodies show the same kinetics of decline as the killing of the virus. So we think this virus was killed by the heat, by flipping the F protein into its non-functional form, so that original vaccine was largely a post-fusion F antigen. So capturing this form, knowing that it flips and loses the potent apical epitopes, and that these induce some neutralizing activity, but not potently, but that keeping it in the pre-fusion form can now elicit responses upwards of 10-fold. Reason for that is because of these epitopes at the apex are more potent. And if you look at a large collection of monoclonal antibodies from both babies and adults and organize them based on potency, high potency meaning less than 0.05 micrograms per mil, most of those high potency antibodies recognize these sites that do not exist on the post-fusion form of F. There are other high potency antibodies, and we'll talk about those, but most of the high potency response to a full F trimer are to these apical epitopes. Just as an example of how this works, MedImmune, before it became AstraZeneca, shared some of their PBMCs from their trial of this post-fusion F molecule so that we could compare those PBMC B-cell responses to pre-fusion immunized subjects. And using probes that are either the pre-F or the post-F on the x-axis, looking at the B-cell response by quadrants after immunization, you can show that in the pre-fusion F immunized, there are pre-fusion exclusive B-cell responses induced. And there are also dual specific antibodies that are recognizing sites on both the pre-fusion and the post-fusion. But in the post-fusion F, only antibodies, mostly only antibodies to the shared surfaces are being expressed. These strips represent mostly a site 4 and a site 2 antibody response, with some site 1 here at the bottom. And these pre-fusion exclusive responses are the ones that engender the greatest potency. So if you look at, again, the collection of monoclonal antibodies, pre-fusion specific antibodies have potency overall of about 10 to 100 times more than the shared surface epitopes. But as you see, there are some shared surface epitopes, primarily to site 4, that will also have some potency. So this is an example of how Pfizer group finally published its exact sequences that it's modified to create its stabilized prefusion F molecule. It uses one of the cavity filling sites that were used by our group, a different disulfide, but linking the same regions of the highly mobile to less mobile areas together to hold them from moving. And also this other site at the base around 486, 7, and 8 that can be modified to stabilize the molecule. And they showed that this is a stable and immunogenic product. We're also going to be talking about antibodies because they're coming along as well. And in addition to the apical antibody, the D25 is the one that's been converted to nirsevimab by AstraZeneca and Sanofi, and it's now well on its way. But there's another antibody, the Merck 1654, that recognizes a site that's mostly site 4, but it does have, so the site 4 in the blue strips here is one of the antigenic sites that Jose Malero originally identified. And then, but it also binds the beta 3-4 hairpin loop on what we call site 5. So this is a quaternary epitope, and it's similar to AM14, which is used to judge the stability of F conformations. And you see in this image, this is the site 4 epitope, this loop over here, but this antibody binds the tip of this loop and the tip of the beta 3-4 hairpin on the other protemer. And so this quaternary epitope, AM14 recognizes it in one way. This Merck antibody comes in from a lower angle, but they're both recognizing this site 4 antibody that's basically a quaternary prefusion specific binding antibody. So this work, the prefusion structure came almost 10 years ago, a little over 10 years ago. It came in 2012, published in 2013, went through all these processes. It took almost 3 years to formulate and get this into a vial, to even start the regulatory process and phase 1 testing, and now advanced clinical trials. And so this is on a more of it's fast, relatively fast historically, but nothing like what happened during coronavirus, but it's finally coming to fruition. And bookending that development process, it was recognized that this is a breakthrough. This is something that's making RSV vaccines feasible, and now we're finally up to the point of having some products. And so, and there's a lot of different products. And so I'm listing the ones here. I'm not listing all the ones in development, ones that have at least initiated phase 3 testing. There are some other products, like GeoVax has a pox virus vector that has been made that expresses F and the N protein that has shown promising results in adult challenge studies, but it hasn't entered phase 3. These are the ones that have entered phase 3, and some of them have now gone all the way to the FDA for a biological license application acceptance. And one of them, the GSK, now has been approved for use. It's also been approved by the EMA in Europe, and this antibody, the nirsevimab antibody that has now been approved in EMA and is on the fast track for review by the FDA in the US. So the antibodies include this site 0 antibody from AstraZeneca and Sanofi, and a site 4 antibody from Merck that is in a phase 3 trial now. And I'm not sure when those results will be available, but big season we had in the Northern Hemisphere in 2022, I'm hoping it will be relatively sooner than later. The vaccines have been developed for the elderly or for maternal use, and they're also slightly different. GSK and Pfizer both have made protein-based vaccines that are stabilized with the fold-on trimerization domain. GSK only uses subtype A with their adjuvant that is similar to what's used in Shingrix. It's just got half of the amount of QS21 and MPL in it is the AS01E instead of AS01B. Pfizer took a different approach. They have used subtype A and B subunit proteins together, but without an adjuvant. Janssen used even a different approach. It was a gene-based delivery using an Ad26 vector, which is membrane anchored, plus at the same time gave a subunit protein, a F prefusion F stabilized subunit protein, and they have a phase 3 trial published. But unfortunately, it sounds like they have withdrawn from development, and all the reasons for that are not clear to me. Moderna also has a gene-based delivery approach that's in what should be a relatively late phase 3 study at this point. It's also a membrane-anchored, stabilized form of the prefusion F. And I just mentioned the Merck antibody that had a phase 3 trial initiated in 2021. So there's a lot of new products coming. We already have the antibody, the ReSViNET antibody, and the GSK elderly vaccine approved in Europe. The GSK elderly vaccine approved S. And the Pfizer maternal vaccine is going to undergo a VRBPAC meeting, an advisory meeting to the FDA on the 18th of May, coming up next week. It'll be reviewing these data that were recently published in the New England Journal showing that immunization of mothers with the subtype A and B antigens elicit responses that result in high antibody transfer to infants and protection against severe disease, 81% or 82% at 90 days, and 69% efficacy at 180 days. And for less severe disease, even 57% efficacy at 90 days. So this is good news. It looks like it's going to be working in maternal setting. If the mother is not immunized, or for either access or for other reasons, the ReSViNET product from AstraZeneca and Sanofi is now available in Europe. Hopefully before next winter season, it may be available. Yes. It is based on this D25 antibody that was originally used to solve this structure. And it also has high levels of efficacy given at birth, even in full-term infants, for medically attended disease. Its efficacy through 150 days is around 80%. And even, and as you look at more severe disease, it's even higher than that, in the range of 86%. So this is also evidence that passively transferred antibody with high neutralizing activity, either from a maternal vaccination or direct delivery to the infant, can prevent severe RSV disease in infants through 6 months of age. And here are the Kaplan-Meier curves, and just to show you that this happened in both the northern and southern hemisphere. It happened in people, in the babies at different ages, both genders. So in all demographics, regardless of weight and regardless of study site, there was efficacy across the board using the nirsevimab antibody. So we have something now for this early risk period of 0 to 6 months. Either direct administration or maternal immunization may be able to cover babies through 5 to 6 months of age. We also now have, these are data from the New England Journal paper of what's now called RSV. It's a clever name. An RSV prefusion F from GSK combined with their AS01E adjuvant. It is having a high level of efficacy against severe disease, and it even reduces disease severity among people who become infected. So this is a respiratory scale severity score showing that even among people who get infected with who've been vaccinated, the disease severity is less than in those who were unvaccinated. And the efficacy is estimated for very severe disease up to 94%, and less severe respiratory tract disease 82%, and milder disease 71%. For the Pfizer protein vaccine that was in the VRBPAC meeting recently, it's a few months behind the GSK vaccine in terms of the regulatory process, but it is also potentially going to be available soon. And that efficacy showed against more severe disease in the 86% range, and for less severe disease in the 66% range. So all of these vaccines and monoclonal antibodies are working either for the 6-month-old child or for the elderly. And the Janssen vaccine that I mentioned was the Ad26 plus F protein bind, also had high level of efficacy, especially against severe disease in the 80% range and less severe disease up to 70%. And it also had good evidence that disease course was less severe in those who were infected and having the vaccine, and their symptom score was less, and they recovered more quickly. And there was some evidence that the durability of immune responses were as good or maybe better than the protein-based approach by itself. So now we have these products that can potentially protect this elderly group. And the question is, what are we going to do for the babies between 6 and 60 months of age who still have some risk of severe disease? It may not be as bad an effect since lung development is more complete by that time, but it still is a place where a lot of hospitalizations occur. And of course, these are the children who spread the virus back to the young babies and also to the elderly, who are going to still be at risk. So I think we still really need an intervention for this. Because of what happened in the 1960s, the regulatory process is going to be complicated. And so most of these approaches, which are felt to be safe, are the ones still in development, either using live attenuated type viruses, chimeric live viruses, or gene-based approaches, either vectors or nucleic acid vaccines, I think would be put into this low-risk category because they're all going to induce CD8 T cells, and in this case, mucosal immune responses, which historically have been thought to protect against some of the aberrant immune responses and causing this allergic inflammation that was associated with the vaccine-enhanced disease. And if they're made with proteins with good, stable conformations, this would be a low-risk approach into that age group. And so we now have these vaccines for the elderly, for maternal, and passing antibody for these young infants. And The question is, how long is it going to take to produce the products for this age group? So solving the structure of the prefusion F revealed this target of vulnerability. We now have proof of concept for structure-based vaccine design, and we have protein-based approaches, vector-based mRNA vaccines that have all been shown effective for the elderly. You have protein-based subunits for maternal and direct administration of antibody. Most of you already know this, but a lot of these, the findings that were extended from RSV are the basis for a lot of what happened during the rapid coronavirus vaccine response. And we think this idea of stabilizing class 1 fusion proteins to make them better vaccine antigens is a generalizable approach that could be applied to other envelope viruses and inform a prototype pathogen approach for pandemic preparedness. So I want to thank especially Jason McClellan and Man Chen and Peter, who all worked to get this early structure and evaluate those earlier constructs, our collaborators in Xiamen Medical University who made the 5C4 antibody, and Tracy Ruckward, who led the team to evaluate all the clinical samples and really do the elegant comparison of post-fusion and pre-fusion F B cell responses. And the PIs I left at the VRC, in my lab that I left at the VRC, moving on to Morehouse School of Medicine, where I really want to emphasize again is my time is going to be focused on technology distribution, manufacturing, distribution and fairness, and equity in vaccine, not just delivery, but equity in vaccine manufacturing and research and availability of technologies for low and middle income countries. And so now that we're having these RSV products, I think most people should be at least spending part of their time figuring out how to make sure these things are distributed in a fair way. I'll stop there and thank you very much.
Thank you, Barney. As always, super interesting presentation. I'm sure there will be a lot of questions in the Q&A in a second, so I'll translate them for you. You can also see them for yourself, but I'll start off. You talked at the end that you're currently working on making the technology available also to parts of the world where the RSV burden is highest. You discussed a lot of different technologies. Can you talk a little bit about what technologies are more easy to transfer to low and middle income countries and what technologies are more difficult to transfer to low and middle income countries to prepare antibodies, mRNA vaccines, subunit vaccines, etc.? Can you talk a little bit about that?
Yes, well, one of the reasons I want to focus on this now is I think there's a new opportunity because of mRNA. And mRNA technology, it's based on chemical synthesis. It doesn't require cell lines. It doesn't require bioreactors. It doesn't require a huge amount of media and a huge campus full of people and equipment. That it changes the equation of what's possible in low and middle income countries. And so mRNA synthesis is something that can be small footprint, small batch. I think it's amenable to transferring that capacity into low and middle income countries, at least initially for research, to build up research programs for regional diseases like Rift Valley fever, Crimean Congo hemorrhagic fever, Lassa, etc., before those things become global threats. And so I just think there's a big opportunity right now in mRNA. And if that kind of thing can be built so that it improves technology and the human capital to do this kind of work, then you can start manufacturing RNA. As that goes, I think that builds the capacity to do GMP production of proteins, which allows you to make now subunit proteins, or maybe the nanoparticles, or the monoclonal antibodies. And this is something that just has to happen so that there's regional distribution of manufacturing capability that can be surge capacity for endemic threats and regional drivers of economies and other things during normal times.
Thank you. It's very clear that we need to prioritize the mRNA vaccines for those parts of the world. So do you have any thoughts on the side effects that we're seeing in the GSK maternal vaccine? Is asked by Javier. An increased risk of premature birth, including mortality. Is there any insight in the underlying mechanism, and does that have any implications for vaccine development in general, Barney?
Yes, well, the GSK maternal vaccine is on pause right now, and I can't really speak very much to that. There has been a public announcement that there was some associated premature birth in that study. In my opinion, it was a pretty subtle signal, but I think that product is on pause, and I can't really say much about it, and the company would have to speak for itself. The Pfizer maternal vaccine, the work that's been published in the New England Journal, and the work that will be reviewed at the VRBPAC committee of the FDA next week, as far as I know, there's no sign or evidence of any kind of prematurity or any other problem in that vaccine. So I'm not aware of anything that would stop it from going forward. But that's based on what I know, and I think we'll just have to wait for that regulatory review. I'm sure the questions are also about the elderly vaccines and safety, and there were some signals in the Pfizer vaccine. There were a couple of demyelinating, a couple, I think 2 or 3 demyelinating conditions in their several tens of thousands of person study, and there were one demyelinating condition in the GSK vaccine study. That's the one that had the adjuvant. So there was actually more in the non-adjuvant than with the adjuvant. So it's something that can happen in trials with very old people and multiple underlying conditions. It's something that was seen during the Shingrix evaluation, and it's such a rare condition that it really cannot be studied very well until the post-marketing phase. Until you get the vaccine into millions of people, it's really going to be hard to even know this is something we should be worried or concerned about. But so I think that these are things that will have to be worked out in a post-marketing setting in phase 4 trials.
You touched upon the adjuvant in some but not all vaccines. Do you think it is important for specific vaccines to add them or to consider adding them? Will it be redundant or have some safety issues? I think this is what a lot of us want to understand for vaccine development. Why is that? And do you have any specific thoughts for the vaccines that are currently under development?
Well, my first recommendation would be to look up all the papers under Dr. Tracy Ruckworth and the evaluation of our phase 1 trial that was published first in 2019. And that analysis, where we used a dose range, but also with or without alum. And it was interesting that alum didn't really add anything in a naive animal. Alum makes it a lot better. But in people who have been primed so many times with live RSV infection, the protein by itself boosts very well. And that's why Pfizer is going forward with it. GSK has a kind of special adjuvant, this AS01E. It may add something more than alum, but in our study, alum was very, very slightly better acutely, but at 1 year, everything was down back the same as in the group without alum. Even a second dose of protein boost made a very slight difference if you boosted at 3 months. But at the end of a year, everything was down about the same. And so the durability, maybe AS01E will change durability. I think that has to be followed over time to see if that's could be the case. But GSK has added the adjuvant. Their efficacy, if anything, was a little bit better than the efficacy in the Pfizer product. And it was interesting because in that study, in the GSK study, 2/3 of the infections in their study were B, subtype B. They only had subtype A in the product. And still the efficacy for severe disease was 94% with the adjuvant, even in a time when most of the outbreak was subtype B. So, you know, because everybody, all the different groups took slightly different approaches to formulation and antigen content, it's going to create a scenario where a lot of interesting things can be studied and learned from these outcomes. So I think there's a lot of work still to do as these things roll out in the post-marketing phase.
It's a relief, at least for me, that there's still a lot to be done in the RSV field. So that's good. Any thoughts on the risk of resistance to the antibodies? Bosco Pace specifically asked about ReSViNET, but I'm sure he also is interested in your thoughts on the other antibodies that are currently under development.
Yes. Well, I think we're going to have an interesting choice to see how this comes out because the maternal vaccine, so, you know, for around 100 micrograms of maternal vaccine, you're going to get about the same outcome as 100 milligrams of antibody given to the baby. And I think there's going to be cultural preferences, whether you immunize the mother or treat the baby. I think there's going to be cost effectiveness questions, and it may be a little more expensive to use the antibody than the vaccine. But some people may rather use the antibody instead of the vaccine. I think antibody production costs are coming down. It's going to be very interesting to see how this works out because both can be effective, and I hope that they're used in conjunction. If women are not immunized prior to birth, the antibodies could save a lot of, they could improve lung health. The antibodies could save a lot of disease, especially if the mothers are not immunized. And in some places, some people may prefer the antibodies. I honestly don't really know what to expect there.
And is there any reason to see that one antibody has a higher threshold for the development of resistance than the other? Is there any, by structure of the binding epitope or the antibody, to be concerned one way or the other based on whatever you know about the structure of the F protein?
Well, the F protein in RSV is not as variable as it is, for instance, that we've seen in the spike protein of coronavirus. And RSV is sort of settled into this pattern where there's two major subtypes. And the F protein of one is, it's got maybe 24 to 25 different amino acids in the ectodomain of the subtype A versus B. But the site 0 antibodies generally work for both. The site 4 antibodies are also very cross-reactive. And the antibodies that the Merck antibody or the nirsevimab antibody cover generally don't change, but they potentially could. If there was enough immunity, it might drive something into a slightly different subtype. There was an interesting study a few years ago using a site 5 antibody that was developed by Regeneron. And that antibody was tested, and it turned out that it was a good, potent antibody, but turned out in the years that they studied it that 2 amino acid changes showed up in the B subtype at 172 and 173. And that was right at the binding site for that antibody. And so it was very effective against the A subtype, but it was not effective at all against the B subtype. So for site 5, maybe other sites, there could be variability sneak in that would make these antibodies less useful, and that's something that's going to have to be tracked. I think RSV has settled into these patterns for such a long time and has always faced so much human pre-existing immunity that I think it's made most of the changes that it's going to be able to make without losing its ability to be transmitted or grow. So in my opinion, it's not likely that these antibodies will be escaped, but because of the Regeneron experience, I think you have to have a little bit of caution.
If you would think about the evolution of RSV over time, then you sort of suggest that there is a more or less stable phase, that the changes that we see now are more subtle than when RSV maybe many years ago developed. But what if we're going to use vaccines and antibodies large scale, also these vaccines, don't you think that the virus is going to adapt to vaccines because of a broad immune response to pre-F, not just shortly in infants, but also in the elderly and pregnant women, etc. So do you think that we're going to see like a fast change in the evolution of the virus?
Well, I don't think so, but I could, I'm sure there's a lot of people with other opinions about that. I mean, RSV has evolved for a very long time in the presence of pre-existing immunity. It continues to reinfect us throughout life. It has had a lot of chances to evolve and change and escape immunity along the way. And so I think that it's probably found that place where it's sufficiently transmissible and grows sufficiently well in human tissue and is able to sufficiently escape most standard immune responses, at least long enough to grow in the nose, that it is kind of in a settled place. And I think it's going to be hard for a vaccine to drive it. What I'm more interested in is whether the introduction of beta coronavirus, a new beta coronavirus now in the seasonal mix of winter viruses, where it's going to end up in the order of, there's a parainfluenza, then RSV, then flu, then human metapneumovirus, and other parainfluenza. And you have this kind of set order of things that usually happens. And if beta coronavirus is put in there in a particular place, how is that going to change the way these other viruses behave? And I think that's all going to be things that have to be studied going forward.
Sonny Kim asked about your thoughts on the GSK Shingrix vaccine and the RSV vaccine, both adjuvanted with AS01. I'm not sure I fully understand the question, but it's obvious something to think about, that GSK has 2 vaccines that both use the adjuvant.
Yes. Well, I mean, the biggest concern, at least a few years ago, was the access to QS21, which is a major component of that adjuvant. And QS21 right now is mostly made from the bark of a particular tree that comes from Chile, the soap bark tree, and it is a limited resource. And so there's efforts, some efforts to synthesize QS21, but it's a complicated molecule. So I'm glad they're using the half dose. AS01E has a half of the QS21 and half of the MPL of the AS01B that's in Shingrix. And so I hope as time goes on, it won't be so hard to manage the QS21 supply. And yeah, so I don't know. Maybe that is her question. I don't think the adjuvants from a Shingrix versus an RSV vaccine, I don't think they will compound the reactogenicity or interfere with each other. But I do think there is a supply chain problem that might come up. Because also the malaria vaccine is using this.
Yeah, so Sonny is indeed adding your concerns about safety, and I think you just addressed that. I just learned that it's apparently easier to make a vaccine than to grow that tree that is supplying the AS01. I didn't know that, so thank you for bringing that up. We're getting close to the hour, Barney, so I want to thank you for presenting and addressing these questions so efficiently. I also want to thank the ReSViNET group that helped prepare this webinar, and everybody who is online. Several hundreds of people are currently participating, and I'm sure on behalf of everybody, I want to thank you specifically, Barney. So have a great day, everybody, wherever you are, and I hope to see you all during our next webinar. Thank you so much.